Hi, my name is Anushka Sanyal and I am from Los Altos in California. Today I will present my work on intronic RNA lariats protect against neurodegenerative disease-related protein aggregation, or put more simply, intronic RNA as a therapeutic target in neurodegenerative diseases, a multi-pronged study of RNA lariat debranching enzyme DBR1. The human and economic impact of neurodegenerative disease is widespread and on the rise. And I see this firsthand as I work with resident seniors with neurodegenerative diseases at a local memory support center. ALS, or amyotrophic lateral sclerosis, is a prevalent neurodegenerative disease that, like many others, is driven by the aggregation of a malfunctioning protein called TDP-43. Recently, the gene for the DBR1 enzyme has been implicated in ALS. The DBR1 enzyme, or lariat debranching enzyme, functions as the only enzyme capable of hydrolyzing a 2 to 5' phosphodiester bond that holds together the structure of looped RNA lariats that are generated through RNA splicing. Specifically for neurodegeneration, the knockdown of the gene for DBR1, which inhibits RNA lariat degradation and thus drives the accumulation of these intronic RNA lariat structures, has been shown to lead to longer lifespans in ALS models. Additionally, DBR1 knockdown reduces TDP-43 toxicity in primary rodent neurons. Unfortunately, DBR1 cannot be knocked out for therapy because its primary function is actually necessary for higher eukaryotes. Additionally, mechanisms through which DBR1 knockdown functions to reduce TDP-43 toxicity are unknown. So I set out to further study DBR1 and, through its inhibition, the mechanisms of RNA lariat accumulation for therapy in yeast. First, I wanted to examine how TDP-43 toxicity is affected in knockout or delta DBR1 mutants. I conducted spotting and competition assays as shown here, and in these spotting assays, the TDP-43 stress cells for the wild type deteriorated at a significantly faster rate than the knockout DBR1. And this was also seen in the competition assays with higher population growth rates for the knockout DBR1. So these tests suggest that delta DBR1 ameliorates TDP-43 toxicity. Next, I quantified how cellular TDP-43 aggregates are impacted by delta DBR1. Mutant cells possessed fewer but larger and more circular aggregates, which were dynamically structured, suggesting a connection to stress granules. Stress granules are non-membranous assemblies of protein and mRNA that localize within a cell and turn static, and this step has been suggested to potentiate neurodegeneration. Finally, till date, delta DBR1 has only ever been implicated in ALS. I wanted to study whether it was more far-reaching. So I conducted a series of assays, first in acute and chronic stress, as shown here, then in intermittent stress, and finally with chaperone systems inhibited. And I concluded that RNA lariat accumulation drives general protection against proteotoxic stress, even when chaperone systems are inhibited. So delta DBR1 must have alternate mechanisms of modifying proteotoxic stress. So overall, intronic RNA lariats alter the property and toxicity of TDP-43 aggregates. Lariat RNA is a general protectant against proteotoxic stress, and the protective effect of lariat RNA is independent from classic chaperone systems. So how can this be used in yeast when a complete loss of DBR1 has adverse effects? Well, currently I'm working on the idea that the role of lariat RNA can be assumed by synthetic lariats, which can be exposed to aggregate-prone neurons to work as a modifier of that proteotoxic stress and then act to rescue neurodegenerative disease toxicity. Thank you so much for listening.